Hello students. In this video, we are continuing the theme of amino acid metabolism. We have some more questions. So the first question is, in the model plant Arabidopsis thaliana, methionine is a precursor amino acid in the biosynthesis of. Then we have four options: alkaloids, glucosinolates, phenolics, and terpenoids. Now to answer this, we'll first start with looking at the structure of methionine. So, what is the unique thing about methionine? It is the presence of sulfur. So, out of these four compounds, one containing the sulfur will be our answers. Now, let's look at all the options one by one. First, alkaloids. So, here is an example of an alkaloid. Morphine. It is a very well known painkiller. So, as we can see, it contains aromatic rings and it is also synthesized from aromatic amino acids. Now on to the second option, glucosinolates. As we can see from this structure, it contains sulfur. So right now we can say that we got our answer, but still for the sake of knowledge, we'll look at the other two options. The third option is phenolics, and a popular example is vanillin, which is the main compound in vanilla extract. Again, we can see it contains aromatic ring and it is synthesized from tyrosine or phenylalanine. And finally, we have terpenoids. So, one example is menthol. Now, as you can see, only one of the options has a sulfur group. So, answer is option 2, glucosinolates. Now, this same question can be asked in another way. Which one of the following compound is not a product of amino acid metabolism? Now, as we just studied, out of all four, the first three, that is morphine, glucosinolates, and vanillin, are made of one or another amino acids. But the fourth compound, menthol, is made of acetyl CoA molecules. So, in this case, the answer will be menthol or terpenes in general. So you can see from this question that same question from the same data can be asked in multiple ways. Now on to the second question. During glycolysis in plants, alanine and related amino acids are directly produced from which one of the following precursors? Our options are first 3-phosphoglycerate, second phosphoenol pyruvate, third pyruvate, and fourth acetyl CoA. Now let's first look at the structure of alanine. Now here is the structure of alanine where the R group in the basic amino acid structure is replaced by a methyl group. Now let's look at the structures of all four options one by one. So first we have 3-phosphoglycerate. Then we have phosphoenol pyruvate. Then we have pyruvate, and at the end we have acetyl CoA. Now take a pause and see that which one resembles alanine the most. Let me know in the comments if you can guess the correct answer. Now, if we add an amine group to pyruvate, we can get alanine. Obviously, it will also undergo reduction, but the closest is pyruvate. So, in this case, the answer will be option 3, pyruvate. Now, on to the third and final question. Aspartate is an amino acid with the following structure. Given are the biosynthetic processes occurring in the cells. A is protein synthesis. B is de novo synthesis of inosine monophosphate and orotic acid. C is synthesis of adenosine monophosphate from inosine monophosphate. And D is glutathione synthesis. Now the question is, which one of the following options correctly represent all the biosynthetic processes wherein aspartate is involved as a precursor? Now our options are: first, only A; second, A, C, and D; third option is A and C only; and fourth option is A, B, and C. Now let's start with looking at aspartate. So aspartate is an acidic amino acid, which means it has an additional carboxylic acid group in its side chain. Now we'll look at all the options. So we'll start with inosine monophosphate and orotic acid. So both of these compounds are purine compounds. And as we can see here that aspartate donates one nitrogen in the formation of purine ring. As we can see here that both inosine monophosphate and adenosine monophosphate contain nitrogens from aspartate. So we can say that both inosine and adenosine use aspartate as a precursor. Now what about orotic acid? So when aspartate is combined with carbamyl phosphate, it leads to the production of N-carbamyl aspartate. When water is removed from this compound, we get dehydroorotate, and after its oxidation, we get orotic acid. Now, orotic acid is an intermediate product in the synthesis of pyrimidine rings. So, again, we can see here that aspartate is also used as a precursor in the synthesis of orotic acid. So, we have covered options A, B, and C. Now, about glutathione. So, glutathione is made up of 3 amino acids: glutamate, cysteine, and glycine. So we can see here that there is no aspartate. Now again, looking at the question, we can see that obviously in protein synthesis aspartate is used. About option B, we can say that it is also used in the synthesis of inosine monophosphate and orotic acid. It is also used in the synthesis of adenosine monophosphate from inosine monophosphate, and it is not used in the synthesis of glutathione. So this is the only odd one out. Now based on this, our answer will be option 4. The correct representation of all the biosynthetic processes is A, B, and C. Now, if you have any doubts in the questions that we discussed in this video, or if you have any doubts regarding amino acid metabolism in general or about a particular amino acid, let me know in the comments. I'll address those doubts in upcoming videos. Thank you for watching.